Alright, so looking at this process of meiosis. Alright, so this gives you kind of an overview of what happens during meiosis 1, and first we'll talk about meiosis 1. So each individual step or stage is presented here. Alright, so we talk about each individual stage by itself, but it's important to note that this is one continuous process. So we're looking at little snippets over time and what's occurring in each step that we have labeled. Alright, so first, interphase. Alright, so during interphase, your nuclear envelope's intact, your chromosomes are relaxed. Alright, you have your centrosomes, and during the S phase of interphase, all of your organelles duplicate, all of your centrosomes duplicate, your chromosomes duplicate. And then looking at what happens as the cell transitions into meiosis. Well, here we have 1, 2, 3, 4, 5, 6 chromosomes. Alright, so the diploid number for the cell is 6. Alright, so that means we have 3 pairs of homologous chromosomes. Alright, so during prophase 1, your homologous chromosomes pair up. Alright, so we call this process of pairing up synapsis. And in fact, you actually have a synaptonyl complex, protein complex, that holds together your homologous chromosomes. Alright, your spindle fibers begin to synthesize your spindle fibers, your centrioles, your centrosome begins to synthesize microtubule spindle fibers. And later on, your nuclear envelope begins to break down, and your spindle fibers begin to attach to your homologous chromosomes. Now, it's also important to note that during prophase, you have a process called crossing over, or recombination. Alright, so this involves interactions between your homologous chromosomes. Alright, so you basically have the exchange of genes, alright, between homologous chromosomes. Alright, so if your, let's say, maternal chromosome has copy of gene A, let's say that the dad's chromosomes homologous pair has a copy of gene A, but it's a different allele represented by lowercase a. These can be exchanged. Alright, so you have the exchange of sections of DNA between your homologous chromosomes Okay. So now, these two homologous chromosomes are no longer identical to one another. Alright, in terms of the sister chromatids are no longer identical. Alright. before you had two copies of the same gene. Homologous recombination took place. You now have two different versions of the gene. Alright, so homologous recombination occurs between homologous chromosomes, not between sister chromatids. Now, the point of exchange between your homologous chromosomes is referred to as the chiasma or chiasmata. Alright, so there's a synaptoneal complex that holds together your homologous chromosomes. Alright, so here you're being shown your homologous chromosomes. Let's say blue is dad, red is mom. Alright, so you have your cohesin proteins that hold together your sister chromatids. Okay. You have a synaptoneal complex that forms between your homologous chromosomes and holds together the homologous chromosomes. All right. Then you'll have regions between your non-sister chromatids. Alright. Well, there's similar gene sequences where you can have crossing over, exchange of genes. Alright. And this crossing over point is the chiasmata. Alright, which is shown here and here, for instance. Alright, so this doesn't necessarily happen at just a single location on a chromosome. It can happen all along your homologous chromosomes. Alright, so here you're being shown the same thing, just in a different way. Alright, you have your two homologous chromosomes held together by your cohesin proteins. Alright, you have the pairing of your homologous chromosomes. You have your synaptoneal complex that forms between them. You have breaks in the DNA between two non-sister chromatids where they exchange and lock together, forming your chiasmata, where segments of genes, your genes get exchanged between non-sister chromatids. Alright, so during metaphase, your homologous chromosomes line up along your little equatorial plate, all right, your cell midline, right? Your centrosomes have migrated to either pole of the cell, all right, so we'll say north and south for this instance, and your spindle fibers have attached to the kinetochores on each homologous chromosome. During Anaphase 1, your homologous chromosomes separate. They begin to move towards opposite ends of the cell towards opposite poles. Later on, telophase. All right, your nuclear envelope may or may not reform. Your chromosomes will relax. Or, depending on the species we're talking about, the cells can transition into meiosis 2 right after. Alright, notice here that at the end of meiosis 1, you have two daughter cells that are haploid. Alright, so recall that our diploid number was 6, while our haploid number is 3. Well, each daughter cell has 1, 2, 3 chromosomes. Alright, so each daughter cell is haploid. Alright, so meiosis 2. Meiosis 2 is pretty much the same thing as mitosis in terms of the events that take place. Okay. The end results are different, alright, comparing mitosis to meiosis, but in terms of meiosis 2 and mitosis, they're very similar. Alright, so notice, we don't have an interphase. Alright, there is no interphase. No interphase. Alright, between meiosis 1 and meiosis 2. Alright, so looking at prophase. Prophase 2. Alright, your nuclear envelope, alright, degenerates. Alright, your spindle fibers are being formed. And your homologous chromosomes will pair up. Well, sorry, you don't have homologous chromosomes. Your chromosomes will basically be randomly distributed throughout the cell. Alright, and your spindle fibers will attach to the kinetochores, okay, on your chromosomes. And they will kind of go through this tug of war process where they're going to eventually be lined up along the middle line of the cell. Alright, so this would take place during Metaphase 2. Alright, so your chromosomes line up along the middle line of the cell. Notice that each sister chromatid has a spindle fiber attached to its kinetochore. Alright, so next, during Anaphase 2, your sister chromatids separate. Alright, at this point in time, each sister chromatid is referred to as an individual chromosome. Alright. During telophase, your nuclear envelope reforms, your chromosomes relax, your spindle fibers degenerate. Alright, and you're left with 4 daughter cells total that are all haploid. Original diploid number was 6. Haploid number is 3. Alright, so comparing mitosis to meiosis. Alright, so mitosis, 1 division. Meiosis, 2 sets of divisions. Alright. The end result of mitosis is that you have daughter cells, 2 daughter cells that are diploid. Alright. The end result of meiosis is that you have 4 haploid cells. Alright, at the end of mitosis, your daughter cells are all identical. And at the end of meiosis, your daughter cells are genetically unique. During mitosis, chromosomes line up along the midline of the cell. During meiosis, alright, you have homologous chromosomes that line up along the midline of the cell during metaphase 1. Metaphase 2, chromosomes line up along the midline of the cell. During meiosis, right, you have homologous chromosomes. Mitosis, you don't have homologous chromosomes pairing up. Meiosis, you have recombination. Mitosis, you don't have recombination. Alright, so this is a nice little summary slide for the events that are different between mitosis and meiosis. Alright, so there are 3 particular events that are very unique to meiosis. Alright, and all of these occur during meiosis 1. Alright, 2 of these occur during prophase 1. Alright, so synapsis of homologous chromosomes and crossing over recombination. All of these occur during prophase 1. Alright, homologous pairs of chromosomes pair up and line up along the metaphase plate during metaphase 1. And then you have separation of homologous chromosomes during anaphase 1. Alright, so your sister chromatids, again, are held together by cohesin proteins, and they stay held together all throughout meiosis 1. Alright, in mitosis, your cohesin proteins are cleaved at the end of metaphase. In meiosis, your cohesins are cleaved at the end of metaphase 2.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.